Hello everyone, welcome back to another class on regeneration biology. So today we will learn about adult stem cell, natural and induced, and their roles during regeneration with some special focus on zebrafish. Okay, so adult stem cell, we have seen it in the very beginning itself that every tissue present in an organism should have some amount of stem cell that is present. However, their load is going to dwindle as you age. As the organism ages, the levels of these adult stem cells start to dwindling, and this dwindling eventually leads to showing signs of old age and say in the skin wrinkles, and the tissue start getting some signs of compromised functioning, and eventually the organ will not be repaired or restored as it should have been done or as it is being done during the young age. So this is what happens when the stock of stem cells or adult stem cell dwindle, and adult stem cells are indeed present. Like you get a new nail that is grown if your nail got damaged, you get a new hair grown if your hair got fallen off, and you get new skin that is formed after a minor cut or a small burn injury, etc. All credit goes to your stem cell, and every organ has got some way or the other some representation of stem cell, and which we call them as adult stem cell. Intestine also we have discussed. Those who have forgotten, you can refer back to the old lectures, there you can see that. So adult stem cells, one of the well studied group is in hematopoiesis. Stem cells on various tissues are essential for tissue turnover and homeostasis. And we know our bone marrow, inside the bone, that is called bone marrow, has got dedicated stem cells that is meant for producing mainly WBC and RBC. Of course, it has got calcium producing cell, fat storing cells also. And aging has both quantitative and qualitative effect on the stem cells. Considering the balance, the qualitative changes of stem cells are more important, because whether they are able to make all cell types as it should be. Say for example, a given stem cell is producing 4 types of cell in 25, 25, 25, 25%, like 1:1:1:1. Say in old age, one of them goes higher, one of them goes lower, then the balance is affected. So it can lead to some complication, which do happen. So not that you have the ability to produce, you should also have the ability to produce adequately or sufficiently. Say you are cooking a food. Normally you take 1 cup of rice, and if you are eating with some curd or something, you will put a pinch of salt. What if you make other way around? 1 pinch of rice and 1 cup full of salt. You are putting the same amount of curd. Will it taste good? Definitely not. So the combination also is important, not that mere presence. Sometimes if the combination has to be 1:1:1:1, it should be maintained. You cannot go that 2:1:1 or 2:1:1:0. It can't happen like that. So this balance is very important for the normal functioning of many cell types. So again, the balance also depends on the availability of stem cell. What is the load of stem cell that is present? So the self-renewal potential of the adult stem cell also is very important, and its developmental potential. That means the ability to give rise to a particular cell type from this cell type, from this stem cell, and the interaction with its ability to interact with its extrinsic signal. For example, hematopoiesis is generally maintained at normal and reduced performance in acutely apparent when old stem cells are subjected to stress. In old age, if the stem cells are also getting equally old as the individual, and if you give some stress, it can be anything, it can be chemical stress, physical stress, some stress, then the cell will not produce as it should have been. The balance will be tilted. Let us see stem cell turnover across mouse tissues. You can see hematopoietic, that is in a dormant state, and this is hematocyte, and epidermis, that is ear epidermis, hematopoietic cell when they are active, and male germ cell, and small intestine, epidermis, and epidermis first is in the tail, then in the paw, and then colon and esophagus. These tissues do have their stem cell. But when you say about the turnover, you see the time in the x-axis, it is the time mentioned in the number of days, 20, 40, 60, 80, 100, 120, 140, 160, like that. As you grow old, even the oldest mice, say 140 or 160 means around 5-month-old mice, means really old mice. The turnover is present in hematopoietic, whereas when around 100 days itself, the hepatocyte turnover is compromised. By around 20-30 days, the epidermis, ear, means 1 month old, the turnover is getting lower and lower. And you can see here some of them, the esophagus, colon, etc. Very early stage itself, the average cycle of stem cell getting shorter. So you can say, as the organism grow older, the turnover of these cells is not holding the ground as it was right in the beginning. At around 0 stage, or maybe first few days, this is 20, so this is first 5 days, the turnover is more or less equal for all cell types, means all are very vigorous, all are quite vigilant. As you grow old, by around 140 days, by 100 days, the hepatocyte disappears. That means 100 to 140 days, the turnover is mainly for the hematopoietic stem cells. Means as the organism grow old, the representation of stem cell, which is reflected in the form of turnover, gets compromised. So this is a fact. So diminished functional capacity of hematopoietic stem cell during aging, which is a fact. Now let us see in this picture in the cartoon form. The red color is young active stem cell, and this purple color is quiescent stem cell, and this one circle with a cut across is apoptotic stem cell. So in a normal scenario, in young age, you can see 4 of them are young active, only 2 of them are quiescent, and only 1 apoptotic cell. And they are differentiating. This blue color one is they are becoming either lymphoid identity or myeloid identity if it is a hematopoietic stem cell. As you grow old, you see this apoptotic cell has doubled, and quite a few of them have already undergone old active stem cell. That number is increasing active, but they are very old. And you can also see the quiescent stem cell number remains more or less the same, and the differentiation is happening as of now. But when you give stress, what happens is the apoptotic stem cell is more. The number 1, which was in young age, now became 4. That means 4 times increase. And although the other type of cells are maintained, the stress in old age becomes a dramatic change in the survival of these young animal to middle age to the old age, which combined with the stress, it makes lot of havoc. And you can also see the restriction in the developmental potency of stem cells during a aging process. Now, restriction basically means during developmental stage, what happens to the potency of stem cell? What affects the stem cell? Of course, we all know that the inner cell mass of the blastula that give rise to a embryonic stem cell, or at a very beginning stage, the zygote is nothing but a totipotent cell. It can give rise to an entire organism, as you can see here. So embryonic stem cell lines derived from the inner cell mass of retained totipotency and can maintained in culture. But if one individual cell, if it is placed in the uterus in the right environment, it can give rise to a entire organism. So that we call it as totipotent cell. And this grown cell, what you are growing, they are coaxed to give rise to other cell type. Let's say hepatocyte, skeletal, endothelial, neuronal, whatnot. You can give rise to any cell type. And hematopoietic stem cells can also be give rise to their routine lymphoid and myeloid lineage. But this can be tweaked by some given conditions. You can also that is called a transdifferentiation. Hematopoietic stem cell that should have give rise to lymphoid or myeloid lineage, but now we can twist the arm or force this stem cell to give rise to hepatocytes, skeletal, endothelial or neuronal cell types also. However, if it grows old, like you can see here in this graph, this is the sharp end shows it is the youngest, and as you grow wider, it is the oldest. As the aging continues, what happens? The hematopoietic stem cells are differentiating, but you may see this is a thin arrow, this is a thick arrow. What does it mean? Hematopoietic stem cell usually used to give rise to lymphoid and myeloid lineage equally. Now the lymphoid lineage is minimalized and myeloid lineage is more. Like in the beginning of the lecture, I told you 1:1 or equal or the proportion that is affected. Same hematopoietic stem cell, it was giving equal lymphoid, equal myeloid, now becoming less of lymphoid, more of myeloid. And most interesting thing is they are not vulnerable for transdifferentiation, means their plasticity is reduced. Same hematopoietic stem cell, just because the cell was older, it is not able to transdifferentiate whatever you do. That means in the young age, these stem cells do not retain the potency to produce a new individual, but they can give rise to any new cell type in young age. However, differentiation of the hematopoietic lineage is skewed toward differentiation at the expense of lymphopoiesis. That means why it is forcing more of the differentiation in old age? Because it do not want to maintain a sturdy population of itself, do not make a copy of itself, and it is now waiting for a chance to just finish off the job. Like when you are, like if I want to give a example, small kids when they are working, they will do very meticulously. They will not do like they enjoy doing that. But once it became a routine thing, one mundane thing, you will just want to see the end point. You just want to finish it off. That is what these cells also do. They just want to differentiate, finish off the job. And it do not explore this trans-differentiation strategy. Even if you pick them out and try to culture and force them, they are just a layback attitude for those cells. Under certain experimental and possibly physiological condition, stem cells in young bone marrow have expanded developmental potential. That's what it has been mentioned here, to produce several other cell types. And although it is not the purpose of this hematopoietic stem cell, but they have the vulnerability or they have the potential. So it is been measured in old age. That vulnerability is lost or maneuverability is lost, and also it do not give rise to lymphoid and myeloid lineage properly. And now if you see further, what is the stem cell response to stress? You can see in this picture that it's an intact tissue and it's a wounded tissue, and how it is responding. We have seen how the wound healing etc. happens and how it is responding to the wound. You can see wound repair in esophageal epithelium. That is what you are seeing here. Following wounding, the progenitor cells next to the wound, immediate neighborhood, exit the cell cycle, that is marked as W. Here you can see here. Exit the cell cycle and migrate towards the defect, that is yellow area, what has been shown in this picture in the inset. Behind this migrating front, cycling progenitors undergo divisions heavily biased to self-duplication. They make a copy of themselves because you need raw material to fill the gap. Cycling progenitors undergo divisions heavily biased to self-duplication, that is shown in the green areas you can see here, and expanding the progenitor compartment to generate the excess cells required to repair the epithelium. This is what has been shown in the cartoon form in this picture. And then what happens in the B, wound repair epidermis? How does it follow? Like you can see here, the thickness has increased in this epidermis. And in the epidermis, the progenitor cells change their behavior as shown in the esophagus. Still, the wound repair supported by the flux of cells into the epidermis from hair follicles, that is the neighborhood cells, purple arrows, what has been shown here in the case of skin epidermis, and the tail skin mobilization of quiescent cells in the epidermis. And the migration explains the appearance of radial clones. That means they'll come in a circular fashion around a healed wound seen in a lineage tracing experiment, as it has been people have followed, where you mark it before and then see where it ends up. That is called a lineage tracing experiment. In the simplistic sense, like if you put an agarbatti in the house, you put the smoke, that smoke is spreading, and wherever that smoke is going, all those places you can get that fragrance. So smoke being opaque, you can watch it, but if that smoke was not there, you will not be able to watch. So that smoke is now acted as a tracer. So this is a simplistic example to follow how cells are moving across in the animal. And the same logic applies if you have got any ablation and repopulation experiments, what has been done in various tissue types. Following ablation, stem cells in the bulge, as you can see here in this. So this is a projection where you have done the ablation wherever you like, and this is an ablated area, and now the stem cell has to come from here and then migrate, colonize, and then fill the gap. That is how the restoring of the normalcy is happened. But however, the contribution of individual cells from the neighborhood is very important. If these cells are stressed, then this orchestration or organization gets affected badly. Those who are interested can read this article that is cited here. So now we will move on to another very interesting organism called zebrafish. So zebrafish is extensively used to study as a model organism because it has got several advantages. One is it has got external fertilization, transparent embryos, and embryos can be injected with RNA, DNA, protein, etc. And it is amenable for large scale genetic screen, forward genetic screen, reverse genetic screen. You can do it and you can study the mutants. And the humans and zebrafish share extensive genomic homology and synteny. Synteny is the order in which the genes are arranged in the chromosome. And zebrafish can regenerate very complex organs such as central nervous system. That is what it makes a very attractive model. And this is a fish which is a typical aquarium fish present in many pet shops. And if you look the brief structure of the zebrafish retina, you can see zebrafish eye in this cartoon. You can see this yellow color is the retina, this blue color is greenish blue color is the lens, and this purple color in the front is the cornea. And you can see there are multiple layers, that is outer nuclear layer, inner nuclear layer, and ganglion cell layer that is present. You can see rods, cones, and also which are basically the photoreceptor layer. And behind the photoreceptor layer, you have got pigment epithelium that is black in color, which attracts the light. And in the middle, the most important cell that is contributing to the retinal function and also the retinal regeneration is the Muller glial cell, as you can see here, which it wraps around every neurons and provides structural and functional support. This is very important for the biology of retina and its function. So if you see the retina, you need to have a proper injury model. Like if you want to study regeneration, one should know how to injure the retina. So we use something called a needle poke method. What is the needle poke method? You take a 30 gauge needle and poke from the back of the eye, as you can see here. In the back of the eye, you poke it, it creates a hole which is uniformly injuring the entire eye, which is easy to perform. Focal injury, so the undamaged neighboring tissue can act as control. Say you have damaged here, so you can see here or here or here or here, anywhere you can see as a control. That means you do not have to study the eye of another fish or another eye of the same fish to study the regenerative response. You do not have to go for another tissue. That is the beauty of focal injury. Although people use other methods such as chemical method, photo bleaching, etc., but you don't have to go behind those methods because they are not uniformly damaging. Light is damaging more of the photoreceptor layer. Sometimes this chemical treatment damage more of the ganglion cell layer, whereas the injury model damages all layers equally. So this is a retina regeneration cartoon in which you can see this is a normal retina and you have got a stab wound, and these Muller glia cells, they enter into cell cycle and they migrate into various retinal layers and restore back the normalcy. So what are these spatial and temporal factors, genetic, epigenetic factor controlling these events? That is a very interesting area of research. You can see here these Muller glia, they dedifferentiate and give rise to pluripotent progenitor cell, and they can give rise to the Muller glia itself, give rise to bipolar cells, horizontal cells, ganglion cells, rod cells, cone cells, and amacrine cells. So this is how the regeneration is restored. There is no presence of dedicated stem cells in the retina. The existing Muller glia de-differentiate to give rise to all the other retinal neurons and the Muller glia itself. Now, the most interesting thing that how it is doing, how is it able to do that? This is something very exciting over so many years. Like you have seen here, you have got the Muller glia and it gave rise to de-differentiated population of cells. Now, who will tell that you need to have this many number only? Say you have got 1000 Muller glia in the retina. How many de-differentiated cells are formed in the eye? How many of them are capable of giving rise to a specific number? Who decides? Of course, we can tell some specific factors, transcription factors are important. You would have also heard about iPSC, induced pluripotent stem cell. However, in retina, no one is there to transfect or no one is there to deliver this protein or these factors into the Muller glia. So the induction has to happen ingeniously or indigenously within the cell, and it cannot go too much. If expression is too much, then it can become too hyperproliferative and you can end up getting reactive gliosis or you can even end up getting tumor. However, this number is fixed. Around 30% undergoes apoptosis at later stage, but it never overproduces. It produces adequate number and give rise to the same set of retinal neurons. It won't happen that there is a too much of abundance of bipolar cell or too much of abundance of rods. It doesn't happen. The same proportion is maintained. And this happens because of the help of lot of pluripotency factors that are induced during retinal regeneration. This is the qPCR data of retina, which is taken from one of my previous paper. Those who are interested can read this article. And this graph shows at various time post-injury, 0 hour, 15 hour post-injury, 2 days post-injury, 4 days post-injury, 6 days post-injury, and 8 days. In this time point, this last point is 7 days, not 8 days. And that is why graph is ending here. You can see lin28, c-MYC, SOX2, NANOC, KLF, OCT4. These are pluripotency factors. Other one important factor is ASCL1A. So there are 6 pluripotency factors, which includes both Thomson and Yamanaka factors. They are induced in this retina at various time post retinal injury. Some of their, say you can see c-MYC B level is very high within 15 hours, and whereas some of them are induced very high at around 2 dPa, 4 dPa. But the take home message is that they are induced significantly in the eye. Now these are able to reprogram the Muller glia. Muller glia is losing its identity. It is now becoming spindle shaped or spherical or spheroid shaped cells that are stem cell. So each of these pluripotency factors have got the ability to reprogram the genome of the Muller glia, and it can give rise to a specific proliferating group of stem cell that is able to differentiate. But you should also understand their level has to come down. Like you can see here, 2 day and 4 day is very important, because in a needle poke injury, 4 day is the peak of proliferation. That means the stem cells or this progenitor cell, retinal progenitor cells derived from the Muller glia, they are maximally expressed at 4 dPa. But see, interestingly, by around 7 day, when the proliferation has come to, or the progenitor proliferation has come to a minimal level, the levels of these genes are decreasing, dwindling. I agree that some of them are staying back. c-MYC level stays high, ASCL1A level still stays high, which is not a pluripotency factor, but it's a proneural gene which pushes the stem cell into neuronal lineage, and which is also important gene required for the formation of an organ called pituitary. All of you would have heard about this pituitary gland. And here the lin28 level also stays high. In this paper, if you read, you will know how ASCL1A regulating lin28, which is a pluripotency factor, and c-MYC also stays high. But whereas others like OCT4 or SOX2 or nano, their level comes down. So what you understand from here, the induction of pluripotency factors are essential for the so-called dedifferentiatability of the Muller glial cell, and that is able to give rise to all the retinal neuron. Now the question is, are they pluripotent? We are saying yes, they are able to give rise to the retinal neurons and the also the glia, Muller glia itself. But do we have some evidence for that? What we did was we made use of a transgenic line. What is that transgenic line? One line is GFAP, which is glial fibrillary acidic protein that is driving GFP. And another line is called a truncated tubulin promoter line. What is the difference between them? The GFAP is turned on in every Muller glia. So you can easily sort them. Every Muller glia will express this GFP under the control of glial fibrillary acidic protein, which is a cytoskeletal protein of the glial cell. Whereas TUBA1 is also part of cytoskeleton. Since it is truncated, it will not express in every neuron or every glia. It rather expresses in those cells which has got some proliferative capacity. So if you injure this TUBA GFP line, you will end up getting proliferating GFP expression only in the proliferating cell. They will have BRDU also, but they will also have the GFP. So you can make use of this line to sort them. As you can see here, this is a cell sorting, fluorescent activated cell sorting done. And you can, in this window, what you are seeing is Muller glia cells. In this window, the middle one is a TUBA GFP, but no injury is given. Hence, no cells are represented. Here, injury is given for DPI. You can see in this window the number of cells that you can see here. These cells can be picked and you can do whatever experiment you want. You can do an RNA sequencing, you can do a microarray, whatever experiment you want you can do. But what we did, those who are interested can read this, my another paper, which is published a few years ago. You can read this paper to know more details about this. What we did was we picked these cells and we thought, can we put it into some embryonic stages or developing embryo? If they are able to give rise to only retinal cell type, they will not get incorporated during development. But if they are able to give rise to other non-retinal or non-eye related genes, then they will be able to give rise to some other tissue type, or at least they can participate in embryogenesis. To test this, what we did was we took these cells and we delivered them. The Muller glia derived progenitors can participate in embryogenesis or not. That was the question. So what we did was we took these cells, as you can see here in this group, in this window, this R3 window, not from here, because these are quiescent Muller glia, that is a normal retina Muller glia. They are not activated, or rather they are not proliferating. They are having GFP expression as a steady state feature because GFAP is a housekeeping gene of the Muller glia. But these ones are activated Muller glia, means they are proliferating, or rather they have all pluripotency inducing factors as I showed in the previous graph. They are now picked. That means from this window, these cells are picked and we delivered into a normal embryo, which is a normal zebrafish embryo, which is developing in the morula stage, say roughly around 6 to 7 hours of development post-fertilization. We delivered. When we allowed them to grow, we have seen that it is contributing. As you can see in this picture, it can give rise to every cell type. We haven't characterized them, but I just want to tell you, these are muscles and also epidermal and various other cell types. As you can see here, this green parts what you are seeing is nothing but these retinal progenitors, Muller glia derived retinal progenitors from the retina is incorporated into, this is the yolk and this is the growing embryo, incorporated, and it was able to colonize. And this is a 4-day old embryo, 4-day post-fertilized embryo. It's 96 hours after you could see this green expressing cells that is present and they are contributing to the embryogenesis. This is something interesting that shows from the retina you can get real pluripotent stem cells. So we'll continue about regeneration biology in another class. Thank you.